Welcome to this educational video on crossing over, one of the most important processes in genetics. Crossing over is a mechanism of genetic recombination that occurs during meiosis, the type of cell division that produces reproductive cells. This process is fundamental to creating genetic diversity in sexually reproducing organisms, making each individual unique. Throughout this video, we'll explore what crossing over is, when and how it happens, and why it's so crucial for life as we know it. Crossing over is the exchange of genetic material between homologous chromosomes during prophase 1 of meiosis. Let me break that down. Homologous chromosomes are pairs of chromosomes that carry the same genes, with one coming from your mother and one from your father. During crossing over, segments of DNA are swapped between these paired chromosomes. This exchange results in new combinations of alleles on each chromosome. Alleles are different versions of the same gene, like different versions of a gene for eye color. This shuffling creates chromosomes that are different from either parent's original chromosomes. The timing of crossing over is very specific. First, it occurs during meiosis 1, which is the first division in the process of creating sex cells or gametes. More precisely, it happens during prophase 1, the initial phase of meiosis 1. The process begins after synapsis, which is when homologous chromosomes pair up and align closely next to each other. This pairing is essential because the chromosomes need to be positioned correctly for segments to be exchanged. Crossing over completes before the chromosomes separate and move to opposite poles of the cell. This timing ensures that the genetic material is properly exchanged before the chromosomes are distributed into different cells. To fully understand crossing over, you need to know 4 key terms. First, homologous chromosomes are chromosome pairs with the same genes but potentially different alleles, with one chromosome from each parent. Second, synapsis is the pairing of homologous chromosomes during prophase 1 of meiosis. Third, chiasmata, which is plural for chiasma, are the physical points where chromosomes cross over and exchange genetic material. You can think of these as the connection points where the swap happens. Fourth, recombinant chromosomes are the chromosomes with new combinations of alleles resulting from crossing over. These are essentially hybrid chromosomes that contain genetic information from both parents mixed together in new ways. Crossing over follows a precise 5-step process. Step 1. Homologous chromosomes pair up during synapsis, aligning themselves side by side. Step 2. These paired chromosomes form a structure called a tetrad, which consists of 4 chromatids, 2 from each chromosome. Step 3. Non-sister chromatids, which are chromatids from different homologous chromosomes, break at corresponding points. It's important that these breaks occur at matching locations. Step 4. DNA segments are exchanged between the broken chromatids, with pieces from one chromosome switching places with pieces from the other. Step 5. The broken ends are repaired by cellular machinery, forming recombinant chromosomes that now carry genetic information from both original chromosomes. This entire process is highly regulated and occurs naturally during every meiotic division. Let's compare what happens before and after crossing over. Before crossing over occurs, you have 2 homologous chromosomes, each consisting of 2 identical sister chromatids. Each chromatid on a chromosome carries the same alleles, meaning they're exact copies of each other. The maternal chromosome has one set of alleles, and the paternal chromosome has another set. After crossing over, the situation changes dramatically. You still have 2 chromosomes, but now they're recombinant chromosomes with new combinations of maternal and paternal alleles. The chromatids are no longer identical to their originals. Instead, each recombinant chromosome contains a unique mix of genetic information from both parents. This mixing is what creates genetic variation in offspring. Crossing over serves 4 critical biological functions. First, it creates genetic diversity by producing unique combinations of alleles in offspring. This means that siblings, except for identical twins, are genetically different from each other. Second, it provides raw material for evolution through natural selection and adaptation. Populations with more genetic variation can better adapt to changing environments. Third, it contributes to population health by reducing the prevalence of harmful recessive traits through new gene combinations. This shuffling can separate harmful mutations from beneficial ones. Fourth, crossing over helps scientists with gene mapping, which is determining the locations of genes on chromosomes. The frequency of crossing over between two genes indicates how far apart they are on a chromosome, providing valuable information for genetic research. Crossing over doesn't happen randomly or constantly. On average, there are 2 to 3 crossover events per chromosome pair during meiosis. This means each pair of homologous chromosomes typically exchanges genetic material at 2 or 3 locations. However, this frequency isn't fixed and depends on several factors. The length of the chromosome plays a role, with longer chromosomes generally having more crossover events. Gene distance also matters because genes that are farther apart on a chromosome are more likely to have a crossover occur between them. Finally, there's species variation, meaning different organisms have different crossing over frequencies based on their biology and evolutionary history. Let's clear up 2 common misconceptions about crossing over. First, people often confuse crossing over with independent assortment, but these are different processes. Crossing over exchanges segments within chromosomes, swapping pieces of DNA between homologous pairs. Independent assortment, on the other hand, randomly distributes whole chromosomes to gametes without any exchange of genetic material. Both create variation, but through different mechanisms. The second misconception involves which chromatids participate in crossing over. Crossing over occurs between non-sister chromatids of homologous chromosomes, not between sister chromatids. This distinction is crucial because sister chromatids are identical copies of each other, so exchanging material between them wouldn't create any genetic variation. Only exchange between non-sister chromatids from different homologous chromosomes produces new genetic combinations. To summarize, crossing over is a fundamental mechanism that shuffles genetic information between homologous chromosomes during meiosis. This process creates genetic diversity that is essential for evolution and species survival. Without crossing over, offspring would only have the combinations of alleles present in their parents' chromosomes, severely limiting genetic variation. Every unique individual, including you, owes their genetic makeup partly to this remarkable process. The specific combination of traits you inherited resulted from countless crossover events that occurred during the formation of your parents' reproductive cells. This elegant molecular dance ensures that each generation brings new genetic combinations, providing the variation that allows species to adapt, evolve, and thrive in changing environments. Thank you for watching this video on crossing over and genetic recombination.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.